And Kate, while I have you, from a QC sampling perspective, what are your thoughts on, approaching QC sampling for thawed products without maybe compromising sterility or reducing the therapeutic dose itself?
Yeah, I think that, one of the challenges that we run into, and, I kind of come from the viewpoint of the world of T cells, is, you need something to be frozen well, you need it to be thawed well, you need to worry about things like cell count, viability, composition of your product, but you also need to be worried about the activity of your product. I think when it comes to QC sampling post-thaw, you need to come down to the minimum set of requirements that makes you comfortable as a company or as a product, that you are able to verify that what should be in there looks exactly the way that it should. And I think that part of that is honestly you getting down to your minimal set of criteria and then working with the agency to ensure that that set of criteria that you have come up with are appropriate and can be done in whichever setting you choose to do your thaw in. So if you're doing a multi-center clinical trial, you need to make sure that everyone who's got their hands on that thawed product can recapitulate your QC sampling time points and methods that you need to have done. So in a way, you need to think about minimal risk to the product itself in terms of contamination or cell loss, but you also need to think about scalability too. If you need to do this in multiple centers, do you have assays or QC sampling that is accessible to all of the centers that you need to be in? Because if you need to try to get a very complex assay out to, 20, 30, 40 centers, that's really going to hold you back. So I think that kind of balancing those things will get you where you need to be. And then understanding if you're doing this in a GMP environment, a non-GMP environment, just a BSC, sort of what's the environment that you're thawing in. But I think really minimalizing contact with the product is always important, because any time that you sample, you do risk contamination. So container closure can be important. That can also help you minimize. So these are all things I think as you develop, you see where your risk points are and you really try to minimize them. So I know that's sort of the 30,000-foot view of it, but I think each product itself is a little bit different. And whether you're
Certainly.
administering that product locally or systemically can also impact how it's thawed and how it's used.
Certainly. I saw Bruno, nodding certainly at everything you had said uh, in your response. And so, Bruno, quickly, I'd like to hear from you too. Your take on the QC sampling for thawed products. Without, like I said, without reducing that therapeutic dose. Any input there?
Yeah, no, I just couldn't agree with Kate more. And so I just wanted to double down and emphasize everything she said, and maybe add the perspective that getting post-thaw QC testing to zero, maybe should be the goal. And I think that through, rigorous application of characterization work and engineering controls as you develop your drug product manufacturing process, including the cryo steps, that can be done. And maybe to just tee off something that we're going to talk later on, I think, Aaron, is that, a little bit of a punchline here is that this post-thaw QC testing becomes even more of an issue as we try to scale the clinical trials, as Kate mentioned, right? So then we get into training of the staff at each one of the sites and reliance on them to do things, according to your SOPs. And once you start to scale, things start to break down pretty quickly there.
Yeah, fair. Absolutely. Pratik, I have a question for you. So I want to talk a little bit about, the scaling up of automated thawing and processing for commercial manufacturing. What would you say are those key challenges? Any thoughts on how to address them?
Again, maybe we can start with the idea that there's a difference in scale from lab to, let's say, GMP or operationalizing a procedure. Whether we're talking about the cryopreservation process itself or other unit operations, there's almost always a difference, right? So that's not just a scale up or scale out, but there's differences in things that are often overlooked: time, temperature. And so these are things that you want to build into your understanding as you take and try to translate what is done in a lab setting into what would be operationalized. And so kind of best practices there maybe pertain to exactly that, thinking about worst case and how you would actually mimic that. So I'm a big proponent of trying to do training runs if you can, or mock runs if you want to call it that, engineering runs in an actual GMP type setting. It doesn't have to be an actual GMP space, but you're trying to do it something like that. to really understand some of those constraints. What are the durations that you see? How long does it take to load up a piece of equipment, to document something? That gives you a good frame of reference to then try to apply and mimic with your lab scale, because oftentimes that's a discrepancy that you see. And if someone does actually take the time to do that, more often than not, they're successful in how they scale out or scale up, because they've already accounted for those subtle differences between a lab and an operational center. So.